Good morning, everyone. Today, we are going to explore an exciting unit. In this unit, we have a total of 13 important topics. Our goal will be to define each of these topics, understand their core concepts, and go through the most essential and frequently asked multiple choice questions, MCQs. These MCQs are particularly important for your license exams, so we'll focus on mastering them. By the end of today's class, you should have a solid understanding of the unit, and feel confident about the MCQs you'll encounter in your exams. Let's dive in. Clinical Biochemistry. Unit 1. 1. Colorimeters. Function. Colorimeters are instruments used to measure the absorbance of light by a sample at a particular wavelength. They are primarily used to determine the concentration of solutes in a solution based on their color. Principle. The instrument works on the principle of the Beer-Lambert law, which states that the absorbance of light by a solution is proportional to the concentration of the solute. Key components: light source, usually a tungsten lamp, monochromator, to isolate specific wavelengths, cuvette, to hold the sample, and photodetector. Applications: used in clinical labs for blood glucose monitoring, urine analysis, and environmental testing for pollutants. VVI points: calibration with known standards, maintaining constant temperature for accurate readings, and the relationship between absorbance and concentration. 2. Water bath. Function. A water bath is a laboratory instrument used to incubate samples in a water-filled container at a constant temperature for various biological, chemical, and physical processes. Types. Fixed temperature water bath, maintains a constant temperature, often used for basic heating purposes. Shaking water bath, includes an agitation mechanism to mix samples while incubating. Applications: DNA extraction, enzyme reactions, where precise temperature control is required, and bacterial culture. VVI points: ensuring water is at the correct level, calibrating the temperature, avoiding contamination of samples, and regular cleaning to prevent microbial growth. 3. Incubator. Function. Incubators provide a controlled environment of temperature, humidity, and sometimes CO2 for the cultivation and growth of microorganisms, cells, or tissues. Key features: temperature control, usually 37 degrees Celsius for human cell cultures, but can range from 20 degrees Celsius to 60 degrees Celsius depending on the application. Humidity and CO2 regulation for specific culture conditions. Applications: used for microbial culture, tissue culture, and enzyme reactions. VVI points: ensure uniform temperature distribution, periodic maintenance, and careful monitoring of contamination risks. Important for microbiology, virology, and cell biology. 4. Centrifuges. Function. Centrifuges separate components of a mixture based on their density by spinning samples at high speeds. Denser components move to the bottom, pellet, while less dense components stay at the top, supernatant. Types. Microcentrifuges, for small volumes, example 1.5 to 2 ml tubes. Tabletop/clinical centrifuges, for larger volumes, example blood separation. Ultracentrifuges, for very high-speed separations, example protein purification. Applications: blood separation, DNA/RNA extraction, protein isolation, and cell fractionation. VVI points: always balance samples to avoid damage, maintain rotor speed settings, and ensure the centrifuge is regularly calibrated for accurate results. 5. Balances. Function. Balances are used to measure the mass of substances with high precision. Types. Analytical balances, very high precision, often used for weighing small amounts of substances, example milligrams. Top-loading balances, less precise but useful for larger quantities. Key features: precision, tare function, to subtract container weight, calibration, and internal draft shields to prevent air currents from affecting measurements. Applications: weighing chemicals, reagents, and biological samples. VVI points, ensure the balance is calibrated frequently, use proper containers, and avoid contamination. 6 pH meter. Function: a pH meter is used to measure the acidity or alkalinity of a solution by detecting the concentration of hydrogen ions, H+. Key components: pH electrode, glass electrode, and reference electrode, which generate a voltage that is proportional to the pH. Calibration: the pH meter must be calibrated using standard buffer solutions, usually pH 4, 7, and 10. Applications: measuring the pH of water, soil, food products, and biological fluids. VVI points: regular calibration, proper cleaning of the electrode, and correct use of buffers. 7 Paper chromatography. Principle: paper chromatography separates components of a mixture based on their solubility and affinity for the stationary phase, paper, and mobile phase, solvent. Types: ascending: the solvent moves up through the paper by capillary action. Descending: the solvent moves downward. Applications: separating plant pigments, amino acids, or drugs. VVI points: selection of the right solvent, avoiding contamination, and accurate measurement of distances for analysis. 8 Electrophoresis. Principle: electrophoresis separates charged particles, example proteins, nucleic acids, under the influence of an electric field. Types: agarose gel electrophoresis, for DNA and RNA analysis. Polyacrylamide gel electrophoresis, page 4 protein separation. Applications: DNA fingerprinting, protein analysis, and forensic investigations. VVI points: correct buffer composition, voltage settings, and gel preparation to ensure clear separation of bands. 9 Spectrophotometer. Function: a spectrophotometer measures the intensity of light absorbed or transmitted by a sample. It is typically used to determine the concentration of substances in solution. Principle: light is passed through a sample, and the absorbance is measured at specific wavelengths. Applications: measuring concentration of DNA, RNA, proteins, and chemicals. VVI points: regular calibration with standards, careful selection of wavelengths, and accurate handling of cuvettes. 10 Flame photometer. Function: a flame photometer measures the concentration of metal ions in a sample by analyzing the characteristic color emitted when the sample is introduced into a flame. Applications: determining levels of sodium, potassium, and calcium in biological fluids and agricultural samples. VVI points: calibration with known standards, maintaining flame consistency, and correct sample preparation. 11 Semi-automatic and automatic analyzers. Function: these instruments are used to automatically or semi-automatically analyze samples, typically for blood chemistry or urine tests. Semi-automatic requires manual input for certain steps, but the analysis process is automated. Automatic, fully automated, including sample loading, analysis, and result output. Applications: clinical diagnostics, pharmaceutical testing, and research. VVI points: proper sample preparation, calibration of machines, and regular maintenance to ensure accuracy. 12 Dispensers and autopipettes. Function: these devices are used for precise transfer and dispensing of liquids, with autopipettes offering adjustable volumes. Applications: molecular biology, example PCR, preparation of solutions, and accurate sample handling in research. VVI points: regular calibration, using proper pipette tips, and avoiding contamination. 13 Polymerase chain reaction, PCR. Principle: PCR amplifies a specific DNA segment by cycling through 3 key steps: denaturation, separating DNA strands, annealing, primers binding to the target sequence, and extension, synthesizing new DNA strands. Components: DNA template, primers, nucleotides, TUC polymerase, and thermal cycler. Applications: cloning, genetic testing, pathogen detection, forensics, and research. VVI points: proper primer design, correct thermal cycler settings, and avoiding contamination.